Hello, everyone, and welcome back to this special lectures for Paleocast. And I am Dr. Alejandro Izquierdo-Lopez. And in the previous lectures, we have seen what happened 500 million years ago during this event known as the Cambrian Explosion. How does the Burgess Shale play a role into understanding the early evolution of animals? And we went through the fossil record at the Burgess Shale to see the origin of groups such as jellyfishes, sponges, Lophotrochozoans, nativerans, and so on. And now we're going to tackle the last group of animals, which are ecdysozoans. Ecdysozoans cover very different animals from nematodes to arthropods or priapulids. And together they basically are related because they are molting animals. So ecdysozoans, every time that they grow, they have to shed their outer skin and they have to produce a new one called the egg disease or a molt. And here you can find, of course, the most abundant group of animals on Earth and most diverse ones, which is arthropods, which incorporates things like spiders, ants, bees, and so on. But now let's first tackle with priapulids. So this is the first group here in the tree that has a fossil record at the Burgess Shale. Today, priapulids, also called penis worms, inhabit our oceans. They are mostly like benthic creatures, and you can see that their body it looks like this. So many of the external features that can be used to recognize a priapulid nowadays, they can also be used to recognize them in the fossil record. So some of these traits that can be seen in fossils from the Burgess Shale are annulation, so this body is annulated in different parts. The proboscis, the proboscis it's an invaginating pharynx that has teeth and it's used to capture animals. So many of these animals are predators and burrowers, as well as a circle of spines. So many groups nowadays of priapulids have spines on their frontal part, as well as the ones from the Burgess Shale. So priapulids have quite a good diversity in the Burgess Shale, and I'm gonna just tackle two species. One of them is Otoya, which shows again you can see in the fossil and the reconstruction some of the traits I mentioned, such as the annulation, the spines on the anterior part, and the proboscis, this pharynx with teeth inside. Otoya is, of course, the most common priapulid at the Burgess Shale, and it's also one of the only animals at the Burgess Shale that we have evidence on what it was eating. This is thanks to the preservation of its gut, and inside the gut you can see pieces of trilobites as well as bivalves and other animals. So it gives us direct evidence on what it was eating. And here you can find another one, for example, Luiselia. You can see here these proboscis with the different teeth and the different structures, the different types of annulation, for example. So Lucella and Otoya are some of the animals, priapulids of the Burgess Shale, and you can see that they are not very different from what we have nowadays. So it's not a group that has changed a lot since the Cambrian period. Now we're going to tackle another group, Panarthropods. Panarthropods today incorporate three main groups: onychophorans, aka velvet worms, which inhabit our tropical regions. They are like slug-like or worm-like animals with feet. We have tardigrades, which are miniaturized animals, arthropods that inhabit the waters, and arthropods that you are probably very familiar with them because they incorporate things again like ants or bees and crustaceans, for example. So, what are some of the traits that can be observed at the Burgess Shale that we can identify that you can use them to identify an arthropod? So some of these traits appear at different parts of the arthropod evolution. And one of the things that encompasses most arthropods or all panarthropods is some type of segmentation. Of course, the segmentation is sometimes internal or can only be seen thanks to the presence of feet, such as in onychophorans or tardigrades, but it's more evident on arthropods. And then as we go closer to arthropods, we have further and further structures that become sclerotized. So they become hardened thanks to the presence of chitin. These structures start as the tips of the claws in velvet worms and water bears, but in arthropods, all the body is fully sclerotized with chitinous structures. So we can understand the evolution of all these traits thanks to the fossil record at the Burgess Shale, which encompass all of these with the exception of water bears. The first group here at the Burgess Shale are Lobopodians and are probably characterized by one of the most famous animals of the Cambrian, Hallucigenia. Hallucigenia was reconstructed as an animal that was basically wandering around the bentos using its spines to walk and was trapping particles using some tentacles on its back. This is the description that appears, for example, in Stephen Jay Gould's book, Wonderful Life. Of course, this animal was shifted upside down, and we realized that the spines were part of its back and that the tentacles were its limbs. But maybe what many of you don't know is that there has been another shift from the anterior to the posterior side. So, before it was thought that this was the cephalon of the animal, was the head, and you can still find that in several reconstructions online. These reconstructions are, of course, wrong. Since 2014, we know that the head is here on this tip. So we shifted that. And this thanks to the work of Jean Bernard Caron and Martin Smith, for example, who discovered teeth and eyes here. This also has become just decayed tissue. So we think that this shape was because there was some tissue here, not very well preserved, making this blob. And nowadays, we think that it looks like that. So this is the latest reconstruction of Hallucigenia with its eyes, the different types of limbs, and ending just in a regular fashion. But this is not the only Lobopodian at the Burgess Shale. We have Ovatiovermis, known from only one or two specimens. And Ovatiovermis has these very elongated limbs with some type of CT here. So we think that this animal was capturing particles from the water column. A similar animal to this one is Collinsovermis, not to confuse it with Collinsomonstrum, which are different animals, all of them Lobopodians. And you can see that it follows a similar pattern. We have the frontal legs with spines to capture particles, and then the dorsal side has very strong spines, such as Hallucigenia. Of course, we think that Lobopodians are either the earliest panarthropods, or they are probably the earliest onychophorans. We don't know exactly. Now we go to Radians. So Radians have already acquired a new trait in arthropod evolution. So not only have sclerotized structures, but their first pair of appendage is fully arthropodized, meaning that it has segments and it's sclerotized. So it's a hard structure with segments. The story starts, of course, with Opabinia. Opabinia is a very strange thing that we cannot say it's an actual Radiodon. It's something before the origin of Radiodons, probably. And this is because of its very strange trunk here that has a claw at the tip. And this is very different from Radiodons nowadays, as we will see. It also has five eyes, and it's a very strange creature overall. So we only have two opabinids, one from the Burgess Shale, one in Utah. So we definitely do not know enough about this group. And the story of Radiodons, of course, continues with Anomalocaris. When Walcott in 1912 made his book on arthropods from the Burgess Shale, he also included other animals, and he described these two things. One of them is Petoia, which was at the time described as a jellyfish, and the other one was Anomalocaris, or the strange shrimp. If you ever eaten a shrimp, you can see you normally eaten the pleon, which is the posterior part of the animal. And this looks like this structure. So Walcott thought maybe this is just the pleon of a shrimp. Of course, now we know that these both structures belong to the same fossil, and in fact, it's part of one composite. So these tails were actually the frontal appendages of Radiodons, and the jellyfish was in fact the mouth. So this has produced two new species. Once we solved this puzzle in the 80s, we uncovered two species. One of them is Peitoia, the other one is Anomalocaris, probably again one of the most famous animals of the Cambrian. You can see the frontal appendages here, as well as these flaps here that used to swim and the tail fan. But Radians have also increased their diversity in the last years. For example, we have Cambrian known for many specimens from the Marble Canyon area, as well as Titanocoris, both of them described by the same authors, Joe Mosiak and Jean Bernard Caron. At first, we thought that maybe Cambrian was a smaller version of Titanocoris and vice versa, but now we think they are both completely different animals. Titanocoris is 50 centimeters. Probably, if we put the body together to the carapace, was around 70 centimeters in size. So one of the largest animals of the Burgess Shale. One difference between these Radians and Anomalocaris or Petoia is that these appendages were not to grasp prey. They were actually like some type of filter feeding appendages. So we think that they were racks that were used to sift through sediment and capture animals or small particles there. So they were not predators in a sense, they were just benthic dwellers. Now we have Stanley Caris. So a series of works by Joe Mosiak in the last years have made this probably the best well-known bradyodont of the Cambrian period. What we knew in 2010 was only this small appendage of Stanley Caris, and it was described as such. But now we have a full view on the body of Stanley Caris. Not only that, but we also have uncovered soft tissues inside the head, so brain tissues of this arthropod, and as well as we can reconstruct the growth series. So because we have specimens of different sizes, we can put them. Sorry. So now we have, for example, Stanley Caris. Stanley Caris is probably the best well-known radiodon from the Cambrian period, thanks to the works of Joe Mosiak in the last years. What we knew first Stanley Caris was only its isolated appendage in 2010, and nowadays we have a full reconstruction of its body. Not only that, but we have also uncovered nervous tissue inside the head, so the brain is preserved, as well as when we put all the different sizes of Stanley Caris together, we can uncover a full description of its growth. Not only that, but Joe also re examined the appendage and discovered that it had features that were not described before, making it a very, very complex functional appendage, probably being able to bite in different directions and different types of prey. And then we have the early era arthropods. So one of them is, of course, Surusicaris, belonging to this group called the Exoxids. So Exoxids is a Bible arthropod that's found all across the Cambrian period, and they are typified by this pair of appendage that looks like a radiodon claw. But they also lack any type of strong segmentation on their bodies, so that's why we think they are quite ancestral animals. Surusicaris only known from one species so far. And then we have Yaunic. Yaunic, of course, is a slightly more modern animal. It has a full body made out of plates and strongly sclerotized appendages. And probably what's more interesting about this animal is its frontal claws, which are made by these three segmented pieces and that terminate in some type of flagellum. So they were used both for predation and also for detection. So they were sensory organs as well. And now we go to trilobites. Of course, the fossil record of trilobites in the Burgess Shale is quite large, but what makes it unique is the presence of soft tissues such as antennae or the limbs or the tail. So this uncovers a little bit more about how our trilobites lived, what type of predation were they doing, and so on. Sorry. And then we have another series of animals very closely related to trilobites, but still very different. One of them, for example, is Tegopelte, which is a very strange arthropod made one of one giant plate with a lot of legs, and Scania, which looks like a trilobite but is, of course, quite distinct. So these animals were probably related to trilobites, but somewhat still different. And of course, we have Naraoya, also closely related to trilobites, but not a trilobite. And the last study by Myers and other colleagues at the Burgess Shale discovered different potential species, including Miss Julia, which was found only in China so far. One of the most charismatic trilobites is Peronopsis. So Peronopsis is made out of like these two very big shields and a couple of segments in between. And Peronopsis in the early 2000s was discovered in Sweden, and the idea was that maybe it's a crustacean. It's just a crustacean that looks like a trilobite. And then Joe Mosiak and Jean-Bernard Caron described some specimens in 2018, and what they found was that it had different types of appendages on its head. So it has the antennae and then five appendages. And this head conformation is closer to trilobites than to crustaceans. So now it's classified as a trilobite again. Another very unique animal and very abundant is Marella. Marella is known from thousands and thousands of specimens, probably one of the most abundant animals at the Burgess Shale. And Marella, what we think is that we have a very good view on the anatomy of Marella. So this reconstruction by Garcia Bellido is really good, but we still are not confident about where does it place. Is it a trilobite? Is it something else? We are not exactly sure because it's such a unique animal. And then we have the history of early chelicerates. Of course, nowadays, chelicerates encompass things like horseshoe crabs, spiders, or scorpions. And the thing that identifies all of them is this pair of small fangs that they have on their heads. So if you look at the spider, you will see two small fangs, and that's how you recognize it's a chelicerate. So Abelia had been known for a long time, but Jean-Bernard Caron and Cedric Aria reinvestigated this animal and found out a very complex head with a lot of appendages. They did not find the fangs, but they found that the configuration of the head was very similar to a spider today. So we think that this was a very, very early chelicerate. And then we change to Mollisonia. Mollisonia has the small appendages here, and these have been described as the small fangs. So we think that Mollisonia was slightly more closer to Chelicerates than Avilia, and is reconstructed as such. It's kind of like a, let's say, the ancestor of spiders, horseshoe crabs, and so on. And now we want to Bible arthropods. So there are many arthropods in the Cambrian that are typified by this carapace. And the carapace is normally quite a hard structure, so it can be sometimes preserved without the body. There's a lot of diversity of Bible arthropods at the Burgess Shale. As you can see, all of them are present. And I'm just going to focus on the on the latest ones. One of them is, for example, Neurocaris, discovered in 2012 by David Leck and Jean-Bernard Caron and other colleagues. And you can see it has this very, very elongated body with a lot of segments and this carapace with a keel here. You can also see the keel. But we also have another more animals that are very similar. One of them is Fibulacaris, which is reconstructed with this spine here going on the ventral side of the animal. So we think that Fibulacaris was probably swimming upside down and was protecting its body thanks to its very long spine. There are many Fibulacaris. There's like 100 or more specimens at the Marble Canyon, and was very abundant, but was maximum 2 centimeters. So pretty small animal, sometimes swimming with a group of Fibulacaris. You can see them all together. So kind of a animal that was going in groups. This Pacucaris is quite different though. Pacucaris is only known by 8 or 9 specimens. And what is very interesting about this animal is this shield-like structure. No, it not only has the frontal shield, but has the posterior shield, which makes it look very similar to a trilobite. So maybe it's some type of conversion evolution. And finally, we have Malhuticaris, also called the sad nun or the beagle of the Cambrian by the internet. And Malhuticaris is around 25 centimeters, the largest specimen, and the smallest one is around 12, as far as I remember. And Balhuticaris is typified, by this very strange carapace that only covers the frontal part of the head. You can see here two Balhuticaris, one here, the other one there, as well as these compound eyes here. So now we have another group of Bible arthropods, which are slightly different. You see, all of them are very similar. They have this very multi-segmented body with very simple limbs. And now we have more complex animals. For example, Tocumia. Tocumia has these very strong appendages here. They look like claws. So it has some type of differentiation in its head. And then we also have Tuzoia. Tuzoia is actually one of the most widespread animals of the Cambrian. It's found from China to Australia to North America. And for a long time, what we have about this animal was just the carapace. And then we revisited the collection last year and we found the limbs and the tail and the head and so on, making this very unique reconstruction of the animal. So we think that's how Tuzoia looks. It's kind of like a predator, very armored predator. So now with that, I concluded all of the animal groups and how the evolution started at the Burgess Shale. Now the question is, are there any animals that we don't know what they are? And the answer to this is yes. So we do have some animals which are completely unrelated to everything else, or at least we have not managed to put them inside another group. For example, Polygeria, which is kind of looks like a sea slug, or something, or like some type of echinoderm, like a holothurian, but we don't have any evidence for that. And we have Prince Cancer marinus, sometimes called a barnacle, but we also think that it doesn't look anything like a barnacle. Then we have these two groups, Deinonychus and Siphosanctum, which have this stalk and this calyx here that they will probably use to filter particles. Maybe some type of sponge, but also completely different from sponges nowadays. And then we also have Portalia. Portalia, you can see here the gut preserved, but everything about its body is kind of diffused. So we don't know if it's an arthropod or if it's a vertebrate. So very, very strange creatures overall. And with that, in orange, you'll find all of the animals found at the Burgess Shale, and everything else is not found there. However, there are only a few that are found outside of the Burgess Shale. For example, Cynorhynchs, probably one in China. We have also some potential bryozoan, one urochordate from Utah, but not much else. In fact, most of this group, we don't have any evidence of their existence during the Cambrian record. We think that they're probably there, it's just that they have not been preserved. So, what are some of the take home messages to understand the diversity of animals at the Burgess Shale? We know that there's a lot of different phyla present, so the Burgess Shale is very rich in the number of body plans we find. However, we do not find all of them. This is why, first of all, some of them may be very difficult to preserve. For example, cycleophorans, which are tiny. Other ones, we don't have traits that can be used to clearly identify them, like platyhelminths, for example. We don't have a clear trait that if we find a platyhelminth in the fossil record, it will be very difficult to convince everyone that it is, in fact, a platyhelminth. But they will also have things that we don't know what they are. So we do have these things that we can have two options. One of them is that they are very strange or ancestral parts of other groups. For example, maybe this is just a strange sponge that sometimes afterwards died. Or actually, they were some type of phyla that were completely different and they would just died out after the Cambrian period. So another thing that is very interesting is the difference between the molecular phylogenies and the fossil record. So, most molecular phylogenies show that many of these groups already were very diversified during the Cambrian period, but this is not what we're finding. We're finding that most of these groups were still in a quite of a primitive stage or making morphologies that afterwards did not continue. This is what we find, for example, of arthropods. So, molecular phylogenies indicate that we should have already horseshoe crabs, and other types of like crabs and lobsters in the Cambrian fossil record, and that's not what we're finding. What we're finding is that these groups were still very primitive. So this gives some type of debate on how are we understanding molecular phylogenies and how do we put the fossil record together. This is an interesting debate that we can have. Another thing that happened is, as I mentioned, is that many of these groups were creating morphologies that afterwards became extinct. And this related to the event that I talked in the first lecture of the Cambrian explosion. So evolution was trying many different morphologies, and of course there has been new morphologies occurring after the Cambrian, but many other morphologies have completely died out. And another thing that I wanted to mention is that we're very happy because we have that every community at the Burgess Shale is quite different. It's depicting a specific time period. Of course, it's always the Cambrian. It's probably always around 506 million years ago, but maybe some of them are slightly different, maybe 507 million years ago or are slightly different. And also, they encompass different localities. So some of them are closer to the submarine cliff I talked about. Other ones were quite far from that. So this creates a better view on the Cambrian period that is not one specific ecosystem. And this is something that it applies to all the Cambrian period. We want more sites that depict other types of ecosystems that we don't have them so far. So if you liked everything about these lectures and you want to give me any comments, any questions, or I missed some things because I cannot control everything, so I may have said something that are not entirely true, or for questions of timing I had to reduce, and maybe sometimes when you reduce things, you oversimplify them, you can write to me at this mail. You can also follow me on social media. I write a lot about popular science topics from arthropods to other zoology and history and so on. And it would be very good if I would be very grateful if you follow me, because that's what I'm trying to create. I'm trying to create a career on science communication. If you want to read more about the Cambrian period, I recommend these papers here. For example, Cambrian Explosion. These ones are very, really good reviews about the Burgess Shale to understand where is it located. The Burgess Shale ROM website, which I took many pictures from. Animal evolution, for example, arthropod evolution, and of course, I also want to thank all the artists that you have seen here. I try to put all the time, but maybe I miss some. These amazing artists have brought the reconstructions to life, and you can see them all spread across um, our presentations. So, with that, I will say thank you to everyone for coming here, and yeah, I hope you got a good time. Thank you very much.